Okay, if everyone's here, I think we'll get started. So it's my sincere pleasure to welcome Dr. Martha Bagnall today as our Neuroscience Center speaker. So a little bit of background. So Dr. Bagnall got her bachelor's degree at Yale University in biology. She then took a position in Thomas Caru's lab as he was transitioning to be the chair of the UC Irvine Neuroscience Department. She worked there for a few years, and then she went to graduate school at UC San Diego, working with Dr. Sasha Dulac at the Salk Institute, studying the role of the cerebellum and the vestibular system in regulating balance, and doing some really great just physiology of those cool cells. After that, she did a postdoc with Massimo Scanziani at UCSD again, working on thalamic inputs into the cortex and looking at how the structure of the synapse regulates the cortical activity. And then she transitioned to a great model system, Zebrafish, where in David McLean's lab at Northwestern University, where she's been kind of continuing studies of balance and so forth using Zebrafish as a model system, as she'll talk about today. So Dr. Bagnall's been doing some really cutting edge work, integrating electrophysiology, behavior, imaging approaches to try to understand how we kind of function in the universe, as it were. She's been receiving great awards along the way. So she was a Pew Scholar, a McKnight Scholar, and an Alfred P. Sloan Fellow as well, since she started in 2015 at WashU, her lab in the Department of Neuroscience, where she's an assistant professor. So today, Dr. Bagnall is going to talk about balance and movement in the larval zebrafish, and I'm pleased to welcome you here today.
Thanks so much for that introduction, Kevin. That was really lovely. And yeah, I got into zebrafish somewhat unexpectedly. I'd actually done my as Kevin was saying, my graduate and first postdoc work in mouse, and got into zebrafish, in fact, because there was sort of a two-body problem, and I was trying to figure out someone to work with, and ended up doing it. And I thought it would be just sort of a side thing that I did for a little while, and then I'd go back to mouse. And now here I am a decade later, still talking about zebrafish. So sometimes the paths that you take on this course is not the direction you expect it to go. All right. So as Kevin said, I'll be talking about circuits underlying balance and locomotion in larval zebrafish. And I'm highlighting today two stories that are particularly about what I think are the things that ended up keeping me in the zebrafish system after a lot of experience in mouse was has a lot to do with the accessibility of the system for many different types of techniques and approaches, in particular in areas of the brain and spinal cord that tend to be less accessible for certain types of mouse and other organism experiments. So I'll try and kind of highlight how I got into that as well. All right. So we'll start by talking about posture. So animals actively maintain posture, and this may seem obvious to you or it may not, but you can think about what happens to posture if you stop maintaining it for a short period of time. This is not a particular situation you have much here in Miami, but generally speaking, the experience of slipping is very disorienting, right? If you stop maintaining posture for a longer period of time, right? And if you stop maintaining your posture permanently. So what this tells you is that all vertebrates effectively, including fish, have to actively organize their behavior to maintain their orientation with respect to gravity. It's not just something that is naturally always the position that they find themselves in, right? And so what the lab is interested in is how you take sensory information from the inner ear and transform that into the circuits that are going to allow you to maintain your orientation with respect to gravity, because it's really vital, right? If you're not oriented properly with respect to gravity, it's hard to locomote, it's hard to do just about anything in this world. So what are the basic circuits underlying this vestibular sensitivity? So this starts in the inner ear, and the mouse here. And it starts out at the two types of sensors in the inner ear. So there's the otoliths, which is the utricle and saccule, and the semicircular canals. The semicircular canals are good for sensing rotational head movement, but they can't tell you anything about your orientation with respect to gravity. So you're really reliant on the otolith organs in order to tell you about your orientation with respect to gravity. And it turns out that in larval zebrafish, we humans and most mammals have utricle and saccule, two organs, but the zebrafish really have to rely just on the utricle because their saccule they use for hearing instead. So it's a neat system because they're completely reliant on this one type of input to get their orientation. And so that gives us some nice chances for kind of manipulation and evaluation of the system. So the utricle sits on top of hair cells that release glutamate onto these vestibular afferents, and specifically utricular afferents, so we're talking about afferents going to the utricle. And these afferents then project axons into the brainstem, where they synapse on a variety of different targets that are involved in controlling a range of behaviors. So startle, postural control, and gaze stabilization type circuits. So it's useful to know a little bit about how this works and how the hair cells here in the periphery function. The hair cells all have the little cilia sticking up into something that's kind of like a mesh right above them. And what that means is that when you tilt your head relative to gravity, the hair cells, the mesh slides on top of the hair cells. And if it slides in the direction of the tallest cilia, that depolarizes the hair cells. And if it slides in the other direction, it hyperpolarizes them. So that means that there's a built-in kind of directional sensor about not just are you tilting your head, but what direction you're tilting it in. And the hair cells are arranged underneath the otolith in a fairly stereotypic pattern that gives you information about a lot of different directions of head tilts. So this is a schematic here as though you were looking down from above at the utricle, which is kind of like a pancake shaped structure. And each of these little arrows, this is just sort of a schematic representation, represents a hair cell in the utricle and its directional tuning. So you have, as you can see, kind of a clear gradient of tuning here, and then there's this line of polarity reversal, and the hair cells on the other side are pointing in the opposite direction. And so between those things, you have cells encoding different types of head tilt information, whether you're tilting rostrally, caudally, ipsilaterally. Here I'm schematizing the utricle on the right side of your head. So most of it is devoted to telling you about tilts to the right, and then a portion of it is also devoted to telling you about tilts to the left. The hair cells here are pretty well organized, pretty easy to understand how this is set up. And the afferents that get input from these hair cells selectively innervate hair cells on one side of that line of polarity reversal or the other. So they're either getting information about head tilts to the right or information about head tilts to the left, but not both. And yet, surprisingly enough, we know almost nothing about the organization of these afferents themselves. So this is kind of a particular challenge in the vestibular system, right? If you're familiar with a variety of other sensory systems, we tend to keep neural information well organized so that the way that it's structured and organized in the periphery, for example, in your retina, is maintained as you go through various levels of the nervous system. But in the case of the vestibular system, there's the reason that we actually don't have a better idea about whether this information is organized in any systematic way at the next stage is that these cell bodies, these neurons, are in a ganglion that's in the adult animal completely encased in bone. There's a little bit of space for the axons to come out, but it's pretty covered in bone. So people who want to record from these neurons to understand their activity have to actually record from these peripheral processes, their axons. And so that doesn't give you much information about organization, right? So I'm going to tell you a bit today in the first half about how this system is organized and then how we were able to use that organization to predict behavior and test it and see that it actually exists. And so the approach that we took here was one that I didn't necessarily think that I would get into as someone who does a lot more on the kind of physiology side, but is serial section electron microscopy. So this is a means of getting an EM level resolution of one zebrafish throughout the entirety of its brain. And this was originally carried out by the Engert and Lichtman labs. And so what you do in order to achieve serial section EM is to take repeated sections of, in this case, is a zebrafish head, and create a map where you've taken images at sort of lower resolution of every single one of those sections, and then go back in at higher resolution to image, in the case that they did here, the brain. But in order to get through all of that territory, they had to image at sort of lower resolution than really gives you information about synaptic connectivity, so the fine processes that are connecting these neurons. However, the nice thing about the way they do this is they keep all of these sections together on a series of plates. So we were actually able to get the sections from this lab and go back in and re-image the areas that we were interested in for understanding the gravity sensing circuit. So what you're looking at here now is the cross section of the larval zebrafish head. This dark gray stuff up here at the top is the brain. And here's the utricle. It's basically like a little rock sitting on top of some hair cells in the ear. This big white space here is the ear. The hair cells are releasing glutamate right onto these utricular afferents that are these bipolar neurons. And the utricular afferents are then sending signals into the brainstem. And if you see sort of a dark L shape, how well is that coming through on the screen? Yeah, you can see sort of that dark L shape. That's the area that we went in and re-imaged at high enough resolution that we could understand the actual synaptic connections in this circuit. Only we did this over hundreds of sections, right? So going rostral caudal, this is one coronal section, but we did this over hundreds of sections. And with that data, then we were able to reconstruct a lot of the gravity sensing circuit in zebrafish at a very high level that could let us visualize, here's how these different types of neurons connect to each other and how they're organized. So just in schematic form then, right, this is the utricle sitting on top of hair cells that signal to these utricular afferents, and they've got a variety of targets in the brainstem. We did look at a bunch of them, but today I'm going to focus on their inputs to the Mauthner cell, which is a major type of reticulospinal neuron that's found in fish and frogs. And it's involved in escape behaviors. And I'll show you I'll show you that in a minute, but first we're going to focus on the organization of these afferents themselves, because as I was saying, we didn't know very much about how they were set up. All right, so now this is going back into, sorry, the little cut off there. So now this is going back into our actual data set. So before I was showing you schematics, and now I'm showing you the actual data. And here, this is an example of a hair cell, just one section of one of those hair cells. And you can see this stash of cilia coming out of the top. The tallest cilium is called kinocilium. It's identifiable. You can see that it's got this special structure. It's a true cilium. The others are not. And so by going back through all of these hair cells, we could reconstruct their directional tuning in order to understand what direction of head movement they're best responsive to. And here's the map that we built out of that. So there's 90 hair cells in here, and I'm color coding them here just based on the angle of head movement that they are best representing. So again, these guys here in the rostral half are best indicating kind of rostral and rightwards tilt. This is all on the right side of the zebrafish. And the ones here in yellowy colors are doing more caudal and rightwards tilt, and the ones in red are going to best signify a tilt in the opposite direction. Okay, so we have all these hair cells, and then on top of that, we also can see what afferents they're connected to. So hair cells release glutamate at these specialized synapses called ribbon synapses, which are readily identifiable because they're these dark protein structures surrounded by vesicles. So we could find all of the ribbon synapses, and it's really easy then to quantify them. It's like, look for the big black blob. And so we knew how strongly each hair cell was connected to each afferent and could then actually make a prediction about the connectivity of all of these afferents. So now we're looking at skeleton reconstructions of all of the afferents. And I should say that part of the reason that we ended up getting as far into this project as we did is that we had all this EM data, and tracing all of these different neurons is a real pain, but it is something you can do from home during a pandemic. So it turned out to be a project that we ended up putting a lot more focus into than I had originally maybe anticipated. All right, so now we're looking down from above at these reconstructed afferents. Each of these large circles on the left here is the soma of the afferent, and then you've got its peripheral process coming out here to where the hair cells are. I'm not showing you the hair cells here just for clarity. And these little circles are where they have their postsynaptic contacts from hair cells. What I hope you can see pretty readily here is that a lot of the organization that we saw at the hair cell level is partially recapitulated here at the afferents. So the rostrally located afferents are going to be encoding rostral head tilt. And we've used the same color code here for the predicted tuning and responses of the afferents as we're doing for the hair cells. And then these caudally located afferents are encoding caudally directed head tilts. The one place that the organization didn't work out so well was those guys on the other side of the line of polarity reversal. So out at the level of the hair cells, right, those are very neatly organized. You can tell where they are. But once you got into the ganglion, they were all kind of mixed in with the other neurons. And you can see that better if we actually just show you, oh, sorry, first of all, this is just that there's a very strong correlation between the rostrocaudal position of a hair cell and the rostrocaudal position of the afferent soma. So showing that that organization is maintained at the next stage of processing. And the issue with the these cells from the afferents that get input from the far side of the line of polarity reversal being mixed in. You can see more readily here where we just made a set of vectors representing all of these utricle afferents and kind of projected it down onto one horizontal plane so you could see them a little more clearly. And again, right, what you see here is the blue ones, the rostral ones are rostral, the yellow ones are caudal, but these red ones from the far side are all kind of mixed in. Okay, so that tells us about the organization of that first stage of processing, which was useful to know that there was some structure to that. But now we wanted to take that into central circuits to understand how this was actually contributing to behavior. And so here we're going to focus specifically on the inputs to the Mauthner cell. I should say that we did also reconstruct all their inputs to a two vestibular-ocular nuclei that are responsible for gaze stabilization, as well as to the vestibulospinal neurons, which are important for postural control. But we knew the least about the input to the Mauthner cell. And the Mauthner cells are interesting because these are kind of command neurons in the brainstem. If you drive an action potential in a Mauthner cell, it will drive an escape behavior response in the fish under all circumstances. This will always work. They have very powerful connections with motor neurons on the opposite side of the body, so it causes a directional escape. So if you activate the right Mauthner, you escape to the left. If you activate the left Mauthner, you escape to the right. They're kind of a specialized version of a reticulospinal pathway that occurs in other animals as well, but they are very large and stereotypical cells, and they get input from a lot of different sensory modalities. So they're probably best characterized for some of their responses to sound. So if you play a sharp sound, the animal will make one of these big escape movements away from it. They also have visual responses. They have tactile responses. Basically anything where you might be getting information that there's a predator coming, these cells are going to get activated and get you away from it. But at the same time, it's pretty expensive to make one of these big escape behaviors. So you kind of, you maybe limit the extent to which you respond to all of these different sensory stimuli. But so they get a lot of different sensory stimuli. And so people have known for quite a while that there are these vestibular, utricular inputs onto the Mauthner, but nobody had actually looked at the organization of those inputs or tried to ask how that might influence behavior. So that's what we decided to do. So here I'm showing you reconstructions now of the Mauthner cells on both the right and the left sides of the hindbrain. If you remember, we're just doing all of our reconstructions on the right, and we could, by looking at the Mauthner cell, reconstructing all of the afferents that are making inputs onto it, we could actually predict what the sensory tuning is of that Mauthner cell. So here what I'm showing you in colors are all of the hair cells that contribute to afferents that then synapse onto the Mauthner cell. And I'm showing you in blue here that the Mauthner cell's overall kind of vector weight is for sort of a rostral ipsy.
Could I just ask a quick clarification question?
Yes.
Are you saying that, so I'm puzzled about this directional orientation that you've mapped onto the EMs insofar as, are you saying that map that you've got there is identical from one fish to the next?
We surely don't know that. This is the big kind of heartbreak of the EM, is that almost all of it at this stage is done with an n = 1.
Right, but then you use an existing block or sections, and the directionality comes from physiology, which is from a different animal, right?
Well, the directionality in this case really comes from the measuring the orientation of the hair cells. So because,
I see. Okay.
Yeah, because the kinocilium's in such that clear location, we can predict then each of these hair cells is connecting to this type of afferent, and they're on into the central circuit. Okay, that is a really clear point. So if I did not make that clearly enough, thank you. So this is then what lets us predict that this should be the tuning of the Mauthner cell. And I'll probably just refer to it as the tuning, but it is true that we can only infer it. Okay, but there was another population of neurons that seemed to be involved in the escape circuit that we found as we were doing these reconstructions. And this was a population of neurons I'm showing you here in red. These are their cell bodies over here at the far side, and they send their axons across where they make synapses onto the Mauthner cell on the other side of the head. And when we reconstructed the inputs onto those commissural neurons, we found that their vectors were all pointing in the opposite direction. So effectively what you could predict then is that the Mauthner cell had tuning in one direction and these commissural neurons had tuning in the exact opposite direction, and they were going to the other side of the Mauthner. Now, because this is EM, we can't prove it for sure, but we think that these commissural neurons are probably glutamatergic, and they excite the Mauthner cell on the other side of the head. And the circuit then that you would kind of predict looks like this. And here I have to pull a little bit of a switch. I've been telling you all along about best responses to head tilt, but the otolith is an inertial sensor, which means if you tilt your head to the right, that'll activate a population of afferents. But that same population of hair cells or afferents will get activated if you translate your head to the left. And that's because the otolith is just sort of an inertial sensor that's sitting on top, and gravity and translation are both accelerative forces, right? So we'll suddenly switch to talking about acceleration. All right, so here's what we think is going on. If you are little zebrafish trying to avoid predation, and a predator is coming at you from the right side, that's going to tend to cause a big movement of water, right? You as a zebrafish are going to get translated over to the left. That's going to activate this ipsilateral tilt pathway, which we think drives then the afferents that synapse onto the ipsilateral Mauthner, the one on the right side of the head here. The right-hand Mauthner causes you to escape to the left. This makes a lot of sense, yeah? So the predator is coming from the right. You're sensing this by the kind of movement of your water column. You're going to want to escape to the left. Conversely, at the same time, though, there's this kind of indirect pathway, right? So if your predator is coming from the left, the head translates to the right, you would activate those few hair cells on the far side of the line of polarity reversal and trigger, then we think, the Mauthner on the other side, getting you to escape again away from the predator. And of course, this circuit is going to be active on both sides of the head, right? But it gives you both a direct and a sort of maybe slightly more indirect set of connections that should let you compute which way are you being unexpectedly accelerated. So we made this prediction looking at the circuit, and then we got back into lab and said, well, let's test it. So here's what we did. We took animals floating around in a dish, and we just had them free swimming because we wanted to, a variety of reasons, one of which being just that it's easier than trying to head embed them so that we had direct control, but it turned out to be pretty useful. So they're free swimming. And what we wanted to do was provide a very strong accelerative stimulus. The reason it had to be strong is that we're just trying to activate vestibular circuits here and not all of the other things. And the Mauthner cell generally isn't going to do its thing and drive escapes just from one modality, right? I was telling you earlier, it's a multimodal cell. It's usually going to be active if it's getting inputs from a whole bunch of different modalities simultaneously, signaling that there's a predator. So we had to give kind of a strong accelerative stimulus to get this to work. And we did this on a sled that we have for a whole bunch of neural recordings as well. And what you're going to see here is that nothing happens at first, and then the Petri dish that the fish is in is going to drift upwards on the screen and then slam back downwards. And when it does, bang, do you see that the fish carried out a kind of classic Mauthner cell mediated type fast escape, where he moved very quickly and made this sort of characteristic C-bend shape, and that's what signifies the startle. Now, is that actually relying on vestibular inputs? Well, we repeated it in a set of animals that don't have the utricular otolith. These are otogelin mutants. We sequenced them a while ago. They were originally developed by Teresa Nicholson's lab. And as you can maybe see here, this fish is actually just kind of light on its side. It doesn't really have these animals can swim, but they don't swim in a consistent orientation with respect to gravity because they don't know which way is up. And we do all of these experiments under infrared light, so they don't even have any visual cues about what's going on, right? So the guy's just kind of hanging out there. And if we give him the same stimulus, what you'll see is that he does a little bit of swimming, but he doesn't do one of those big rapid fire escapes. And that was fairly typical across our population as a whole. So here now is quantification. If we looked at the quantified by our accelerometer here, the stimulus that we're giving to these animals, and then the tail angle response, which we're using some automatic tracking in order to keep track of, we can see that the wild type or heterozygous siblings escape at about twice the rate as these rock solo transgenics. So we think that there's probably some lateral line, so some somatosensory input that some of these vestibular deficient animals are getting that's still causing some escapes, and that probably contributes. But overall, it's clear the vestibular system is an important component in driving these. But then here was the thing about the direction. So they were free swimming animals, right? So we weren't controlling what angle their head was at when we started the stimulus. And what that meant was that we could look at escape trials and ask, what direction did they escape relative to the stimulus that we provided? So the x-axis here now is the animal's initial heading angle at the point that we started giving the acceleration. And the acceleration is sort of in the downward direction here. So this animals here under 90 degrees, these are being accelerated to their left sides. Animals at 270 are being accelerated purely to their right. And what you can see is that animals tended to escape in the direction that we predicted. If they're accelerated to their left, they escape left. If they're accelerated to their right, they escape right. And the outliers that we had that didn't neatly fall into these categories are animals that were being accelerated more or less perfectly rostrally or caudally. They didn't really have a lot of information about whether it was a rightward or leftward. So this was exciting because it kind of validated the reconstructions that we had done from the circuitry. And in fact, then we can repeat that kind of analysis in those vestibular deficient animals. I should say it's a little dodgy in some respects because the animals are kind of lying on their sides. So I'm not sure they're invariably being or driven in exactly the same ways. But at the same time, you don't see any real, in the remaining escapes that they do have, you don't see much response. Yeah. Yeah. Yeah, I'll repeat the question. Okay. Yeah. Yeah. So it sometimes doesn't show up great in videos because they're getting, played through a 30 hertz feedback here. But in essence, if you look at the acceleration profile, that moment where you change direction is a moment of maximal acceleration in that kind of, in that case, downwards on the screen direction. The animals are just moving laterally, right? But we're videoing them from above. And so because it's a very rapid change in the direction of movement, that's the big accelerative force. We've now repeated this experiment because we were kind of wondering, how much did it matter that they kind of did this? We've now repeated this experiment in animals where we just gave them a unidirectional stimulus. So they just got accelerated in one direction, although it does mean they get decelerated, right? Which is like negative acceleration. And we got very much the same response there. Yeah. So we could use that as kind of our sticker. But we did have to do, part of the reason we had designed the stimulus like that was because we were trying to keep the animals within the field of view of our camera. Then we, after a while now we've actually changed it. So we have the entire camera moving along with the fish. So that makes these experiments a little bit easier. But that's why we were doing it originally. Okay, so then what I've told you then for this first half of the talk then is that we can trace an organization of the vestibular system going all the way from the hair cells to the afferents, demonstrating that the afferent ganglion is organized in the rostrocaudal axis by tuning, and then using that to predict computations in the Mauthner cell escape circuit, which we then were able to test with behavior and show really did allow these animals to have a directional computation of how they were being accelerated. All right, so in the second half, then I'm going to move on to spinal circuits. I'm okay to hold questions to the end, but if you've got anything very burning now that you'd like to talk up, I can also take that now. No? All right, let's go on to the second part. You can ask me. Oh, and I should say briefly that I don't have time to talk about this in as much depth, but another component of the organization that we saw seems to be sort of a temporal patterning set up by developmental sequence. We know that the hair cells right around the line of polarity reversal have sort of special encoding properties for fast movement, and they develop first. And we found that they were preferentially connected to early developing afferents, and those early developing afferents preferentially went to things like the Mauthner circuit, which is also very early developing. So this was interesting because there's generally this notion that in motor circuits, there's a development is kind of patterned by this temporal sequence. So you get development of the fastest circuits and then development of progressively finer circuits. This may sound a little counterintuitive, but think of it as the increased development of a later development of fine controlled movements, right? Precision movements. Whereas early, if you consider like how a baby moves, right? There's a lot of just kind of flapping around. They can't do little careful pinches. So what we're suggesting here is that it looks pretty similar in the sensory system, going from hair cells that develop early and seem to encode, or kind of the onset or phasic encoding seems to be driving an early pathway, and then later developing, we think, goes to a later developing circuit. So this is all on bioRxiv, so if you're interested in looking into that, we can talk about that later. Okay. In the second part of the talk, I want to talk about the organization now of circuits in the spinal cord and how we think those are contributing to behavior. So specifically, and this is probably pretty intuitive to you, that there's the rostrocaudal axis of your body is something that's very important for locomotion. So when a fish is swimming forward, right, there's a propagating wave that moves down the spinal cord. You can see that really clearly in salamanders or lizards, something like that, because they still have this axial bend and the limbs are basically moving along with it. And then in more vertical or later developing vertebrates, you don't tend to move the spinal cord as much, but you still have that exact same pattern of limb activation, right? Where your forelimb moves forward, you swing your arm forward at the same time that you're stepping your right foot forward. So even though we don't need the arms for locomotion any longer, you can still see that kind of propagation of activity in the spinal cord as you walk. Yes, you see this in a lot of animals. And here's the example. This is, I'll be telling you about work mostly done by my postdoc, Mohini Sengupta, a fantastic researcher in the lab and also very coordinated in the rostrocaudal axis. Okay, so the key thing that we were interested in was sort of coordination along this long axis of the spinal cord. And this gets into questions about the organization of spinal circuits generally, and part of, again, kind of the reasons to be in zebrafish. So as a piece of background here, the spinal cord is composed of a really diverse population of different neurons. So there's motor neurons that are going out directly to control the muscles, but they make up maybe 5 to 10% of the spinal cord, depending on where you are. The rest of the neurons in the spinal cord are a very diverse population of what's generally just referred to as interneurons or spinal neurons. They're excitatory, they're inhibitory, they go commissurally, they go ipsilaterally. There's a wide degree of diversity here, but excitingly, we've gotten in the last couple of decades a number of genetic tools that give us handles on specific subpopulations of these neurons. And the genetically defined populations tend to have consistent characteristics. So for example, this V1 population is defined by the transcription factor engrailed. These are inhibitory neurons and they project ipsilaterally and ascending axons. And that's true in zebrafish, and it's true in chickens, and it's true in mouse. So it seems like these are consistent themes about the organization of all these premotor circuits. But what we don't have a great handle on is actually how these circuits are contributing to behavior, and specifically their kind of interconnectivity. Now, we know that a lot of neurons in the spinal cord send long-range connections, and that these long-range connections are really important for that kind of rostrocaudal coordination that I was mentioning. So this is an example from Sylvia Arber's lab. If you kill off specifically neurons that have their cell bodies in the rostral spinal cord, this is in mouse, and they're sending their axons to the lumbar spinal cord, you get periods of kind of inappropriate limb alternation because there's less coordination along the rostral caudal axis. So we know these long projections are really important, and yet they're very difficult to study how they're actually working in the circuit. And the reason for this is that, generally speaking, if you're interested in circuitry, you might be doing, say, synaptic physiology to record these connections. And to do that, you've got to cut slices all along the spinal cord. And once you cut slices, you no longer have these nice long connections. So it's actually been a real challenge to understand organizations. So I'm going to tell you a bit about how these V1 neurons send differential connections along the length of the spinal cord and how we think that's important for function. And then a second population of inhibitory neurons that sends inhibitory inputs in the opposite direction, and why we think these two end up having very different consequences for behavior. Okay, I just told you this, larval zebrafish, they're great for spinal cord research, a lot of homology, all of these populations we can find in the zebrafish now. So let's focus specifically on this V1 population. They're defined by the transcription factor engrailed 1. And this is now part of the joy of zebrafish, is you can just look at these in vivo. So here's most of the zebrafish on the top, and then under fluorescence, you can see these neurons all the way down the length of the spinal cord. There's also a little expression out in the muscle here, but that's not important for our experiments. And so we can label these neurons. We can characterize their projections just to see how much they did tend to project. So here we can do sparse label by embryo injection of plasmids so that we can get just 1 or 2 neurons labeled at a time and characterize their anatomy. This is a V1 neuron. Now we're in, I think, just about all of these images, we're looking at these laterally here. So here's its soma, and then it's sending a long ascending axon, in this case for about 4 or 5 segments. Segments are muscle segments in the zebrafish, and they're basically equivalent to vertebrae. And then a shorter axon descending as well in this case. They send they sent these long axons for their connection structures on an average of about 6 segments. And so we started looking at their connectivity. To do this, we took advantage of optogenetics. So we put the optogenetic activator channel rhodopsin into these neurons so that they would be activated specifically when we shine blue light on them. And the a general challenge in using this kind of approach for mapping is that a lot of these neurons are usually activated if you also shine light on their axons. But we managed to find a way to kind of selectively and provide very focal stimulation using a digital micromirror device that let us put stimulation over just a very narrow range of the spinal cord. And when we did that, we found that if we illuminate the area that we're recording from, we could elicit spikes there, but we can't elicit spikes if we go rostrally or caudally to the specific area that we're recording from. So what that tells us is that our little square of blue light is only activating neurons directly under it, not all along the spinal cord. Okay, so that's the tool that we're going to use now to probe the connectivity of these neurons along the spinal cord. It's quantification of that. So the way that Mohini was doing this, which she would record from different potential targets of V1 neurons in the spinal cord, and then move this light up and down the spinal cord. And here we really got to the kind of the advantages of zebrafish, and that because they're transparent, we could do this without having to do a major kind of surgical intervention, and we're doing this all in vivo. So that makes it a lot easier to be confident that the circuit is intact. All right, so here she's recording from a motor neuron. And if she activates V1 neurons in the segment next door, she gets all of these nice synaptic currents. These are IPSCs, or inhibitory postsynaptic currents, showing a strong connection. But when she started activating V1 neurons further away, she got, generally speaking, kind of nothing. And this came as kind of a surprise to us. We knew that V1 neurons were important for inhibiting motor neurons, and we knew that V1 neurons were sending long axons. So what was going on here? Maybe it was just that weird one. No, she looked at those across a whole bunch of neurons. And she saw this pattern of connectivity where if she were activating segments nearby, she could get robust synaptic responses, but as soon as she got a bit further away, no responses at all. So we thought, okay, well, maybe this was because she was targeting these recordings to fast motor neurons, and maybe it's going to be specific to slow motor neurons. So she repeated it in slow motor neurons, and it ended up looking honestly pretty similar. There was a little bit more range of connectivity, but still, as soon as you got 5 segments away, no connections at all. And so this puzzled us, and Mohini spent a while trying many different postsynaptic targets in the spinal cord and kind of getting the same result over and over, at a point that we started thinking, maybe we're interpreting our data the wrong way. Maybe it's this, maybe it's that, and just sort of scratching our heads more and more. And then she went after a different target in the dorsal horn of the spinal cord. So the dorsal horn of the spinal cord, it tends to be responsible for sensory inputs and for evaluating, responding to sensory inputs. And she looked at a particular population that we think is equivalent to what's known in mouse as DI5. These are sensory neurons. And now all of a sudden we could see robust inhibition of these neurons at long distances. It wasn't just the case, though, that everything in the dorsal horn, all of the sensory neurons, got a ton of long range inhibition, because you could record from a different type of cell that got nothing at all in the dorsal horn. So it seemed like there was actually very targeted information and inhibition coming from these V1 neurons onto different populations of neurons. So now I'm going to show you the result of many months of data kind of all collapsed together here in a heat map that reflects connectivity as kind of viewed from the perspective of a V1 neuron, right? So she did all these recordings of the postsynaptic neurons, but now let's think about it from the standpoint of a V1 neuron. The x-axis here is the rostrocaudal distance along the spinal cord. So again, and now that we were able to kind of normalize our synaptic responses in order to compare them across neurons. So these are 2 types of motor neurons, the fast and slow, and Mohini had very strong connectivity locally, but nothing at long ranges from the V1 neurons. Pretty similar results looking at a couple of different populations of premotor neurons that are known to be in the ventral horn and responsible for some kinds of locomotion. But when we include then the sensory neurons in the dorsal horn, you see a really different pattern, right? With inhibition maxing out 3 or 4 segments away and kind of tapering off from that. So what this suggested to us then was that these neurons are involved in inhibiting motor circuits very locally, but sensory circuits at long ranges. This came as kind of a surprise to us, and it's hard. What we would love to have done would be try to manipulate the circuit in a way to change where the synapses were happening, right? To try to understand what the habits to locomotion. We don't have the tools to do that right now. We don't actually know what the molecular cues are that are driving synaptic connections locally versus long range. So we couldn't come up with a way to do that. So instead, we took a reasonable approach here, and that is we built a computational model in order to think about what might be going on here. And so we did a very simple model that just had a couple of different types of neurons in it. And here we had first, we set it up with the V1 neurons inhibiting motor circuits locally, which is exactly what we had observed in our actual recordings. And you can see this nice propagation. This is now raster plots of motor neurons going from rostral to caudal. And you can see there's this nice propagation of activity going along the fish, which is exactly what you'd expect to see in fish. It's just sort of propagation from rostral to caudal. Sorry, there we go. Spikes going along. But if we did the same thing, the exact same model, but now we just moved the synaptic connections from V1 neurons further away so that they're inhibiting at long ranges rather than locally, we got all of this aberrant spiking. So it seemed like there'd be a burst of motor neuron spiking, but then sometimes a weird spike happens after that. And keep in mind that if a motor neuron fires, that's going to cause a muscle twitch. So we predicted then that that's going to have a very deleterious effect on the normal propagation of locomotor behavior. And we can kind of quantify the extent of that, the additional kind of bump of activity there. You can see that it really suggests that a normal function then of V1 neurons is to keep motor neurons from firing right after they've had a burst of activity, right? So the motor neurons are all having a burst of activity in order to drive muscular contraction. But after that, it's not just important that they burst, it's equally important that they shut up after they burst. And the V1 neurons, if you manipulate the location of their circuits here, we think that inhibition is no longer coming at the right time to shut down that motor neuron activity. Okay, so that's pretty much what that is. They're inhibiting motor circuits locally and sensory circuits long range. I'm going to tell you just very briefly about one other population of inhibitory neurons in the spinal cord, because it turns out you got really different consequences of changing this inhibition. I think that's useful to think about. So I told you about the V1 population, and now we're going to talk briefly about this V2B population. The one thing that we already knew about the V1s and the V2Bs is they seem to be really important for flexor-extensor alternation. And this has a lot to do with how we generally think about behavior, locomotor behavior, is we think of it as being composed of different types of alternation. So you alternate between the left and right sides of your body as you walk, and you also alternate between flexors and extensors, right, in a stance and swing cycle. And the Martin Golding's lab had done a very nice job of showing that these V1 and V2B neurons in mouse are important for flexor-extensor alternation. If you lose both V1 and V2B neurons, the mouse limb just kind of sticks out like this. And they can't really alternate their muscles very well. But here's the weird thing. We're working in fish, and yet these V2B neurons are found, sorry, that's the, these V2B neurons are found, and the V1 neurons, as I just showed you, throughout the entire spinal cord in fish. So we know that they didn't start from an evolutionary perspective. They didn't launch their lives as being involved in limb control. They have to have some kind of role in locomotor control generally. And this is just sort of the schematic version of what I was just telling you. Okay, so again, these V2B neurons, you can now see them distributed all the way down the spinal cord in what is obviously a limbless animal. So we decided to ask what their anatomy was and what they might be good for, and try to see what we could understand about the relationship of V1 and V2B inhibition. So V2B neurons, here's a soma here, and you can see its axon going descending. As you will probably notice here, right, the V1s all had ascending axons, the V2Bs have descending axons. I'll give this sort of characterization of subtypes of V2Bs. Then we decided to manipulate them relative to behavior. And we did this in a couple of different ways. If we activated the V2Bs with channel rhodopsin, we could slow down locomotion. So these are now ventral root recordings of motor neuron activity, so that you can see this is a paralyzed animal, but you can see when it's kind of trying to swim, and black is under normal conditions and blue is when we are selectively activating the V2Bs, and there's an increase in the interburst interval, so they're slowing down. And so that's schematized here for data across a lot of fish. Conversely, if we shut down the V2B neurons, you speed up locomotion. So here we're validating a different type of optogenetic construct that we were using, ACR, and showing that it shuts down the neurons when we use it. And now we actually used, for various reasons, we used actual tail movements in these animals. And what we saw is the animals still moved their tail the same kind of amplitude, but they moved it faster if we were shutting down V2B neurons. So this kind of suggested the V2B neurons are functioning as brakes on the locomotion, right? So we activate them, things slow down, you suppress them, things speed up. And part of what was a real surprise here was that's exactly opposite from what happens if you get rid of V1 neurons. This has now been shown in both mouse and zebrafish, pretty consistent results that if you kill off the V1 neurons in the spinal cord, everything slows down. So, right, you lose V2B neurons, swim speeds up, you lose V1 neurons, and swim slows down. Why are these so different? Well, we thought one clue might be by looking at connectivity from the V2B neurons. So Mohini kind of repeated all those experiments that I showed you before, but now in the V2B population. And here what you can see is they have pretty long range connectivity onto motor circuits. They don't have any connectivity with the sensory circuits, and they have instead long range connections down the spinal cord. So what we think is going on here, and this is still at sort of our best level of kind of putting these data together, is that the different direction that inhibition is traveling relative to that propagating wave of locomotion moving down the spinal cord is what's responsible for the differential effects. So if you think about a wave of activity moving down the spinal cord, a V1 neuron that's, we know from our data now, is only inhibiting motor neurons locally, it's going to be activated around the same time as that motor neuron, which means that its inhibition onto that motor neuron is going to come towards the end of the burst, when it would be good for suppressing activity at that aberrant activity at the end, right? Conversely, a V2B neuron is going to be activated at the same time as this wave of locomotion passing down the spinal cord. And so the inhibition that it provides is going to be racing along with that wave of activation, and we think is going to be coming in the early portions of the burst. So it might be more important at turning up or down the gain of the motor neurons so that they're firing more or fewer spikes. So these are very different ways of controlling motor neuron activity with inhibition. And then finally, we just thought it was very interesting that from this two sets of mapping, we did see, if you specifically look at their reciprocal connections, right? V2B neurons here do directly inhibit V1 neurons that are a couple of segments away, and V1 neurons in turn inhibit V2B neurons that are a couple of segments away, which means that there's effectively a reciprocal inhibitory circuit already built in at the level of fish. And that reciprocal inhibition, we think, could form the basis, particularly if you just turned up the strength of those synapses a little bit later, for flexor extensor alternation. And the reason that that works is reciprocal inhibition is obviously a good means of getting two circuits to alternate. And specifically, we know that the motor neurons underlying flexors tend to be located a little rostrally to the motor neurons underlying extensors. And in fact, you can record them at different roots. So there's actually a rostrocaudal difference between both flexors and extensor motor pools, and the V1 and V2B neurons are kind of well placed by this reciprocal inhibition to be important in enforcing their alternation. So that's what we think is likely to be, perhaps there's a sort of original or ancestral version of inhibition that's involved in controlling the speed of locomotion, but it got co-opted later on for limb control. All right, so this just summarizes what I was telling you in that last portion then, that we think that although the traditional focus is on the roles of inhibition in enforcing alternation between left and right, between flexors and extensors, we think that it's got actually original and probably continued functions in controlling speed and other aspects of movement, and that those functions then got co-opted in order to produce some types of flexor extensor alternation later developing animals. And with that, I want to thank the members of my lab. Jikai Liu did the first half of the study that I was telling you about. He's now a postdoc. It's really fantastic help in the reconstructions and behavior from Nick Sliman and Yizhen Jia. Mohini, as I told you, did almost all of the V1 data that I showed you about with contributions from Rebecca and Fabsi on the other elements of the V2B story. And I'm grateful also to our collaborators who helped with all of the EM that I was showing you, and of course the funding sources. And with that, I'd be happy to take your questions. Thank you.
Our Zoom audience, do you have any questions from Zoom? You can unmute and speak.
Yeah.
So what do you think is just you can't move them, force them, I guess, but do you think proprioception also kind of plays a role in this, or do you not even need it because you've got all the inhibition excitation you need locally to kind of account for that?
Yeah, this is a great question. And so proprioception, right, the sense of how your muscles are moving is pretty fundamental to a lot of how animals perceive what they're doing. And for a long time, the assumption has been that it's not relevant in the zebrafish because they don't seem to have the classic proprioceptors, which are like these spindle receptors out in the muscle that let you sense, oh, my muscle is extended or flexed. And they're a lot of what you use to know, like, are you standing, right? Fish don't have spindles. So the assumption was, well, they're not really doing any proprioception. And the last few years, there's been some pushback on that, not necessarily with the notion of spindles embedded in the muscles, but with a couple of different types of neurons in the spinal cord. So there's one population of ciliated neurons that actually sticks up into the central canal. And Claire Wyatt's lab has done a lot of work showing that these CSF contacting neurons are really responsive if you even do a passive deflection. So they think that what's going on is if you bend, the CSF, the cerebrospinal fluid, starts moving, right? And that's going to activate these ciliated neurons that are sticking right up into the canal, and they in turn seem to have some effects on circuitry. And then there's a second population that Abdel El-Banira's lab has identified of these long, they call them something like intraspinal neurons or something, I can't remember the exact name, but they go into the bony junction, or the maybe cartilaginous junction, collagen junction between two bones, between every set of vertebrae. And so they think that they're actually sensing stretch of the spinal column itself. They don't show up till a little bit later. So the fictive swimming that we can do here when we paralyze the animals and try to analyze how the circuit works, it works pretty well, and it largely recapitulates what swimming looks like in vivo, but it's probably not 100% there. It might be 80% there. And how that, and so Claire's lab has shown something about how these CSF neurons feed back into the circuit. It's still not super clear what that actually does for locomotion.
Are there more students? Since I'm not in the room, I can't tell.
There's one, yep.
Just for clarification, when you talked about the direct and indirect circuit, you said they were both glutamatergic?
We think it is. So those neurons that go across the midline and are commissural, we think that they're glutamatergic because it would make sense. But we actually have a really challenging time figuring out how to prove this, because those are neurons that we found in serial electron microscopy. We don't have a great way of going back into an in vivo animal and picking out just those neurons. There's tons of commissural glutamatergic and glycinergic neurons there. And so we're not sure how to find those guys again in vivo.
Should there be also like a GABAergic pathway, like to inhibit?
Could there be a GABAergic pathway?
Yeah, like just to have inhibition also centrally.
Yeah, so I think that there's probably, yeah, right. Could there also be a GABAergic pathway that would go indirectly, maybe from the afferents the other side? I think that is very likely to be in the auditory system, which is where people have done a little bit more work on looking at these commissural connections. There's both glutamatergic and inhibitory, like mixed glycinergic GABAergic, or maybe just glycinergic neurons going across the midline that they think are doing a sort of comparison between the sound levels on the two sides. Because again, if you hear a noise and you're trying to escape, which side to escape to? Well, you want to escape away, but how do you know? From any one ear just has the information about how loud is the sound here, right? So you kind of need to compare between the two ears in order to figure out which way to go. And so commissural inhibition seems to be pretty important in that circuit. So you would predict, I mean, vestibular system's right next door, that we might also see some inhibitory neurons that would be important for this, and we just don't have great ways right now of looking at. I mean, we found some commissural going neurons in the EM data set that don't seem to necessarily be participating directly in this, so they might be doing some indirect inhibition. I think we need to get into a bunch of physiology to really figure that out.
Okay, thank you.
Yeah.
We have a question from Zoom.
Zoom maybe, or
Any other questions? Zoom listeners?
Yes, I think there are several hands up from the Zoom listeners. So I'm wondering if the lateral line afferents actually plug into the same, do they actually input into the same V1, V2, and then sort of have a coordinated, maybe integrated response to the motor system, or is there a separate? And then actually a related question is, different fish use the lateral line system very differently, right? So if it's mildly electrically sensitive fish versus, and they use different parts of that lateral line. So how does that work? And also the utricle is a single organ, and so you can sort of disperse that information to the premotor system, but the lateral line afferents are coming in every segment.
Yeah, okay, so a bunch of things in there. So the lateral line, right, just for anyone who isn't familiar with it, is kind of this system of hair cells all the way down the length of the body of the fish, and it responds primarily to movement of the water, although it's definitely the case it's got more complicated roles in some other circuit and some other fish as well. And the lateral line hair cells, these neuromasts, they synapse onto afferents that are kind of like those utricular afferents, in that they've got their cell bodies in a ganglion that's up in the ear, and they sense input and then they project centrally. And they do project directly onto the Mauthner cell as well, which is why we think that they may be important in driving some of that residual escape in the behavior that we saw in the rock solo animals. I don't think that any of those afferents go to the spinal cord to directly interface with locomotor. I think it's all going to be carried out through the brainstem, and the brainstem is just such a challenge because it's a real mess, right? And so what I don't know is if there are, I assume that there are other target neurons besides the Mauthner that are located in the brainstem that are getting lateral line input and project to the spinal cord, but I know less about their identity. Actually, I'm not even sure how many. They might be going to the rostral spinal cord because there's vestibulospinal populations, but I don't know if there are lateral line to spinal populations. So I don't know how that might be influencing. We also think, I think that a lot of times we think of the lateral line system as more kind of a generalized go signal, right? So if animals feel that they're drifting backwards in a current, they'll just swim and they'll try to orient themselves relative to that current. So we know, I think that they're comparing the left and right sides to do that, but I don't know how that computation happens. I don't know if I covered enough of the.
No, that's fine. I just wanted to know if it was integrated. It sounds like it's not. Yeah, that's fine.
Okay. Dr. Dahlman has a
Okay, that was a beautiful seminar. Thank you. I was curious about the connectivity, the sensory component of the connectivity of the V1 inhibitory interneurons, because those sensory cells also have axons that extend several segments, if I remember properly. And so I was wondering, it was kind of cool to see the gradient of inhibition with distance. And I was wondering about how you think those sensory neurons are integrating inputs from those from the many inhibitory neurons that are presumably interacting with them.
Yeah, exactly. So, so that's right, that these coponeurons do kind of have long processes and long axons. We think that what's going on here, so our best, why have this kind of inhibition of the sensory neurons at all, and also why have it over such long ranges? Because it's completely right that both the V1 inhibition onto them is spread out over long distances, and the coponeurons themselves have really long dendrites. So our best guess here is that this circuit and some work from other labs suggest that what happens with these coponeurons, they're somatosensory kinds of neurons. So they're going to respond to there being sort of like a touch on the skin or something like that. And what seems to go on is that there's sensory gating going on during activity. So if you're walking along and your hand brushes against something, you're not that surprised by it. But if you're just sitting in your chair and something brushes on your hand, right, you're going to react to that pretty strongly. And so there seems to be a general kind of turn down of the gain of the sensory responses during locomotion in a way that it lets the animal say, okay, a lot of this movement that I might be feeling is probably just because I'm locomoting, right? I'm swimming through water, so I'm not too surprised that there's maybe a little bit of a touch. Maybe some touch systems are getting activated a lot. So we think that might be why it doesn't have to be spatially specific, is if front part of the animal's moving, the back part is too, right? There's not, you don't need the spatial specificity in the same way.
Okay, great, thanks.
I was just wondering for labeling the cell essentially, if like does retrograde antegrade labeling work for like if you targeted the ciliated cells, would it label the neurons or is it not compatible?
Oh yeah, so you can do really nice retrograde labeling. And in fact, the picture, I didn't say it, but the picture that was up at the screen on the very start of my talk was retrogradely labeled neurons from the spinal cord, which you can see really nicely. But it won't work for the hair cells if it's just a regular dye, because it won't go across the synaptic junction. So we can't say label from the afferents and try to get it into the hair cells. What we would need for that would be viral tracing techniques. And there have been a lot of challenges getting those to work in zebrafish. There seem to be some promising approaches coming from a few labs now that would let you specifically label things going across synaptic junctions, but it's still maybe in the kind of squishy getting developed phase. But that would really be great for a nice label.
Thank you.
I had a question. Thank you very much for that really wonderful lecture. I thought it was really interesting. I have a question about the Mauthner cell. So it's a cell that's responsible for a reflexive initiation of escape in the fish. I can imagine that you showed an example where you modified the pressure, I guess a pressure wave, and it initiates a movement of the animal. But escaping from a predator is going to be a probably a very violent type of event. And I guess, am I correct in saying that this Mauthner cell and the activity through this circuit is going to be continually activating the escape response? Is that correct?
Yeah, so the Mauthner cell drives an escape. Are you asking, does that escape then activate the Mauthner again, or?
Right, during that sort of encounter.
Yeah, right, because then you have a really sudden
continually
head movement, right? So you might think that then you'd get it kind of going in a nonstop loop. Yeah, so this is a great question. It turns out that the Mauthner has a population of potassium channels, so that it's almost impossible to fire a second action potential in the Mauthner right after you've had one. There's also some inhibitory feedback loops, but mostly it's just that once you spike, you activate this overwhelming population of potassium channels. So the Mauthner cell is very reluctant to be activated again within the next second or so. So it can't then kind of auto respond, right, and get itself into.
So how does the fish normally swim? What's the initiation?
Oh yeah, let's try. Turns out this is a very difficult question to answer. We think that the locomotion is generally initiated by dopamine and serotonin seeming to cause kind of a wave of just generalized depolarization. Maybe glutamate is also in there too. It's a little bit exactly unclear how you can get, if you just take like an isolated fish spinal cord and dump on NMDA or dopamine or serotonin, you can get swimming. So we think that that's how it's generally being initiated. It's been a little bit difficult to test, but there's some people now looking at some of the new like dopamine indicators in order to try to figure that out. But it seems like once you increase excitability in the spinal cord network, just generally, it's self-organizing, and that'll turn into locomotion. So you don't need to trigger this big command response in order to do normal locomotion.
Right. So the fish have a mesencephalic locomotor region, or?
Yeah, where is the MLR and the zebrafish? So the closest thing that people think might be kind of MLR related is what's called the nucleus of the medial longitudinal fascicle, or NUC-MLF. And if you activate those neurons, you can trigger locomotion. It's not totally clear to me at a genetic level if it really corresponds to the MLR.
Right. Thanks.
Yep. Great. Thanks, everyone.